Today we will be talking about the characteristics and the definition of the topic enzymes. So first is these are biological catalysts. What I mean by biological catalysts, there are catalysts that are used in factories to speed up the reaction. So what is the difference between a human catalyst and a factory catalyst? The difference is that these are highly specific. If we put this enzyme in a factory or in an industry or in any chemical reaction, it won't speed up the reaction. So they are biological catalysts. They are not used in any kind of industry, textile industry or any chemical and medicine, any industry. So which alters the speed of chemical reaction. If a chemical reaction is going to take 2 hours, it's going to make it 2 minutes or even to 20 seconds. It speed up the chemical reaction without getting consumed in them. So it didn't get consumed. If this is an enzyme, a protein comes over here and attaches to it, this forms enzyme substrate complex. Then this forms amino acids because protein breaks into two products. These are smaller products which are known as amino acids. So enzyme is not consumed in this process without getting consumed. What about the speed of reaction? I will explain it more. For example, you eat a tub biscuit. It is going to take you like 2 minutes, 20 minutes to digest. But if you eat without an enzyme, it's going to take you 72 hours to digest a single small tub biscuit. 72 hours. It's also possible that without enzymes a body can survive, but it is possible that it can survive, but to speed up those reactions, like if you eat, your body needs quickly and protein and diet. So you eat a tub biscuit, it needs quickly the protein, the carbohydrates, the energy, but it's taking 72 hours. How that cell can survive without food? So it's out of common sense that enzymes are really necessary for your body. I can accept that body can survive and digest the protein, the carbohydrates, the lipids by, you can say, hydrolysis, heat, stomach acid. What about stomach acid? If we talk about stomach acid contain pepsin which digests the protein, protease, trypsin. If these are not present in the stomach acid, how can stomach can digest? It can digest, but it is going to take 72 hours to digest just a single tub biscuit. So if we move on further, now we are going to talk about the characteristics of an enzyme. So first of all, what is enzyme made of? So what is enzyme made of? Enzyme is made of protein. We all studied that protein is the building block of enzyme. That's not a lie, that's a true. So protein, a group of amino acids forms protein and protein forms enzymes. So we say enzyme has proteinous nature. If we talk about the second thing, what is their structure, what they look like? Enzymes don't have a specific shape, they have shapes according to their substrate. What is substrate? The reactant in which they are going to form enzyme substrate complex, that's all. So we will say they have granular structure. Why we say they have granular structure when they don't have a proper structure? If we look under a microscope, these are enzymes. These are looking like small grains, like wheat grains, like rice grains, like any salt grains, any sugar grains. These are looking like grains. So scientists call them enzymes have granular structure. So do you understand why they are proteinous in nature? Because they are made of protein. They are called granular structure because they look like grains under a microscope, because they don't have a proper structure. So if we jump on the third topic, the third is totally dedicated to the point of its structure. This is a normal enzyme. And if we see this, they have two parts. This part and this part. And this part. So if we talk about this part, this is structural part. If a substrate, like such as this is a protease enzyme, and a protein comes and attaches to it, on this structural side, nothing will happen, no process, until or unless it attaches to this part. This is a lipase enzyme, this is lipid. Lipid want to break down into fats. So it's come over here, then it will be break down into the products, not here. If it attaches on this structural side, no process. This structural side is known as allosteric side. The examiner, the CI examination wants us to know proper and biological names for these structures. So this is allosteric side. If we talk about this functional side, where the functions happen, where the procedure happens, this is known as active side. Or the action side, there are two names for it. So if we start writing, enzymes have two parts. Enzymes have two parts. The first, structure, where a substrate attaches, there will be no process, no function. Structural side, which is known as allosteric side. If we talk about the second is known as active side. The functional side, which is active side, where a substrate attaches to it. I hope you all understand this thing. If we move on to the further talks, we will see the point like enzymes. I hope you all have listened this thing in your notebooks, in your books, whatever books or whichever teachers you studies from. They all say enzymes are highly sensitive to temperature and pH. They never told you what is enzyme made of. This is an enzyme made of proteins. So what is keeping these proteins together? The hydrogen bonds, the H bonds are keeping them together. When we give them temperature, these hydrogen bonds break down. That's why the protein breaks down and enzymes demonstrate. So I will be not taking this any further because this is the topic of another chapter. So if we say the specific thing, enzymes are highly specific in the substrate selection. Why I am saying they are highly specific in their substrate selection? I means that this is an enzyme for protein. This have this specific shape for protein. This is a protein enzyme, a substrate of protein, whereas this is a lipid. So this lipid, if it attaches to this side, it's not possible, but if it attaches, there will be no process, lipid won't be breakdown, because enzymes are highly specific. When this protein comes, they form this structure, and then it will be break down into smaller products, product 1, product 2, which is what you all say, amino acids. So they are highly specific. If a substrate, if there's highly specific in this substrate selection, if a substrate, for example, what you say, a substrate like lipid, a substrate like protease, a protein attaches to this lipase or protease, it will only accept for which substrate the enzyme is for. This enzyme is for protein, it will only accept protein in it. It won't accept lipid. It won't break lipid into smaller pieces. So if we move on further, now we talk about the process. Let's talk about the process. When enzyme attaches to the substrate, enzyme substrate complex is formed. What I mean by this thing, if I'm saying when enzymes attaches to the substrate, this is a protease enzyme, this enzyme attaches to its substrate protein. When these two comes together like this is known as enzyme substrate complex. Let's go through all the points once again. Enzymes have proteinous nature. Why they have proteinous nature? Because they are made of protein. Enzymes have granular structure because they look like grains under a microscope. The enzymes are made of two parts. The first part is known as structural part, which is just for structure. Any enzyme come on it, any substrate comes on it, no process. That's called allosteric site. The second site is called active site, where a substrate comes and the process happens. Enzyme is highly sensitive to temperature and pH because they have hydrogen bonds. If the temperature is high, if the pH is high even, or if the concentration is low whatsoever, these hydrogen bonds breaks. When we talk about enzymes are highly specific in the substrate selection, because if any substrate of a lipid, a substrate that is lipid, a enzyme that is protease, that lipids substrate cannot attaches to the protease enzyme because protease enzyme is specifically for protein. When enzymes attaches to the substrate, enzyme substrate complex is formed. When these two, the substrate and the enzymes attaches together, this structure is known as enzyme substrate complex. So thank you for watching my video, guys. So bye.